Let's talk comb jellies. So they're in the phylum Ctenophora. The C is silent. And the teno part means comb, anytime you see that coming up in a name. Phora is like carrying pharaoh, basically. So they're the comb bearers. There are two big classes of these. The Nuda just look like a little sack with the eight little comb rows on the sides, and Tentaculata have these sort of tentacles that hang down with little sort of spiral loops on there. They lack the sort of poison tipped darts that cnidocytes and cnidocysts of actual cnidarians, things like corals and jellyfish, what you would normally consider a jellyfish. Um, yeah, and they just sort of catch things because they get tangled up in these little sticky tentacles. There we go. Yeah, found worldwide, quite invasive. When these things get into the Black Sea, they sort of decimate fish populations, and they're really hard to control. No natural predators, as far as I'm aware. Yeah, and all the modern ones have eight, count them, eight comb rows. The symmetry is going to be bilateral, basically, but it'll kind of look like a circle in some cases. And usually they have eight of almost everything except the stuff right at the top, like a little status list. So here you go. This is a typical one. I believe this is a pleurobrachia. And yeah, it's got a little pass through gut, and then it's got some canals for digestion. They're calling them paragastric canals. In some organisms, these things run kind of underneath the comb rows. And so each part of the animal is doing a little bit to help with digestion. There's a little statocyst, which is down here, and it's got a little, basically a little mineralized stone of some sort. And when that bumps into the side, the jelly knows that it is tilted, so it's a tilt sensor. Um, Here with the phone. So let's say I tilt this, or here, tilt it that way, and then bam, it's going to be running into the side. Yeah, that's the idea. So, that's a ctenophore. Here's some beautiful pictures of ctenophores. And the one thing we're super sure about is that these are very primitive metazoan animals. That's Metazoa. It means like multi-celled animals. Yeah, higher animals, some people would call them, although, higher than what? Everything that exists now has been evolving for the same amount of time because everything shares a common ancestor. So, higher and lower are pretty subjective, I gotta say. Although, as humans, we do usually put ourselves in the highest of high categories, so why not? Yeah. Intelligence has its perks. All right. The genomes of ctenophores are starting to be known. This is from a recent paper, one of several genomes that's out now, and there are several transcriptomes out, like, I don't know, 6 or 7 that I know of, probably more. And so what we can see here is a little ribbon diagram. It's like a tanglegram. And so it's showing the layout of different chromosomes. And these are chromosomes you would find in a type of jellyfish, Rhopalima. Branchiostoma floridae is a little tiny, it looks like a fish. It's a little very primitive chordate. And here's a sponge down here. Iphidatia, and here's a different kind of sponge. And from the look of it, I would say that's probably a hexactinellid sponge. And so what you can see here is mapping genes. And so if there's a gene on this chromosome and it also maps to this chromosome, then they're going to mark them, and they'll connect them with a little line. And that line will be a particular color depending on which sort of group of genes we're talking about. And so you can see between the porifera, pretty much whatever's on this chromosome is on the same chromosome up there. So, chromosome 16 is chromosome 16, 11 is 11, 1 is 1, and there really aren't any genes that have moved around onto it or off of it. When we compare porifera, the somewhere very near the bottom of the animal phylogenetic tree to a bilaterian animal, a very primitive chordate, or an early branching chordate, let's say, then what we see is that, yeah, still a lot of those are the same. Now, we have some wholesale movements, like chromosome 5 over here turns into chromosome 17, but everything that was on 5 shows up in 17. So it's just, a little swap. It's not, it's more the numbers have changed than anything else has changed. And same thing here, comparing a bilaterian to a cnidarian, which are things like corals and sea anemones and jellyfish. And you can see, yeah, like, look at that one over there. Look at these 3 first chromosomes. There are a few genes that have migrated during some of these changes, but in general, they're all pretty close. So from this, what you would take away is say, oh, which are the 2 that are closest to each other? This is probably the 2 sponges. Who's next closest? Like, the sponges are pretty close to the cnidarians. Cnidarians are fairly close to the little bilaterian branchiostoma. And then compare that to the 2 ctenophores on here. And the 2 ctenophores are actually very similar to each other. So they contain genes that normally are found on, like, chromosome 14, mixed with genes that are normally found on chromosome 13 and 18 and 12, depending which organism we are. And they're mixed together in the same way. And yeah, they're on the same chromosome in these. So the 2 ctenophores are very close to each other. And from all the crisscrossing you see in this level, you can see that they are completely differently organized from the other animals. So ctenophores are animals because they have the same genes as animals, but ctenophores have a unique arrangement of those genes on their chromosomes, and it's shared with ctenophores, and it is not found really in any other branch of life. So that makes ctenophores a little difficult to place. Either these are highly divergent, something has happened to them that led to their genomes being really scrambled and then staying the same forever, essentially, or they were a very early branch and something happened to their genomes, or they reflect an earlier version of the genome, and everything else that's alive is basically the other type of genome that was around. Think the Ediacaran would be roughly the time, or maybe just a little bit before the Ediacaran. Yeah, late Proterozoic. So there you go. So we know ctenophores are old. Yeah, the question is how old? How far back can you find fossil evidence? Because genetic evidence is good, but when you're dealing with a weird case like this, then genetic evidence can potentially be a little bit misleading depending on which parts of the genetic evidence you are looking at, and what sort of ways you're using to look at them. Because like a normal phylogenetic tree, you would just align each of these genes, and you might say that, oh, they align, they're very close, and ctenophores might be at the bottom, or they might be near the bottom, they might be before or after sponges. But if you look at the organization and take that into account as well, then probably you put them somewhere, yeah, probably they're a sister group to the rest of animals. That's the idea. All right, anyway, here is an early ctenophore fossil, and this one actually kind of looks like ctenophores now. This is from the Hunsrück shale, which, do you remember which one that is? That's the one where you get pyrite casts of soft-bodied organisms, and you can x-ray them, and they look beautiful. And so here is a fossil, and they have x-rayed it, and you can see it's got little tentacles here. I don't know if that's a tentacle of another one or what, but it should be right there in the fossil, really, but it's not. And here you can see you got this little organization at the top. You can count the little comb rows. There's one going down the back, and I see 7 around the front, so that makes 8. This is a modern-looking ctenophore. And then you look further back, and you see things, and boy, are these ctenophores? Are these not ctenophores? What are these things? What the heck are these things? For example, here, and we have one of these in the teaching collection. Actually, it looks a lot like that. We have something that had hard parts. Now, this is from Chengjiang, so the hard parts are replaced, but occasionally you'll get a little bit of phosphate, which is making up the minerals that replaced them. So the idea is that whatever part was there may have had some extra phosphate in it compared to the rest of the body. And we also know that some really early Cnidarian fossils, like among the first ones to make a like sort of an armored living case, the mineral that they use is calcium phosphate, which is like the mineral in our bones and is probably homologous with that, because these are essentially our ancestors or are a sister group with our ancestors, but real close still. But yeah, you got this sort of prong at the top, and you got a number of comb rows that'll come down the side. And in this case, the number is 8. So, that makes a lot of sense. Or at least there's eightfold symmetry on the top. This is one where the top piece got fossilized in an upright organization. Yeah, and I think there's 4 the main sort of spikes coming down, but each of those has little spines on it, which we'll see in some of the later ones. Yeah, here we go. This is a relative, different kind, but you can see that the armored parts coming off that apical structure, whatever we want to call that, are going to have spines on the outside of them. So these are going to keep other animals back. Yeah, this is probably either anti-parasite or anti-predator. It's a little tough, but paleoecology will probably eventually reconstruct that satisfactorily. You can see here up at the top, the SL, I believe, is a little statolith, which it's a fossil of a thing that was actually mineral at the time. Pretty cool. And you can see the little comb rows running down the side. You can see that 8 is a common number, but as we'll see in a minute, 8 is definitely not the only number. You also get multiples of 8. There's a 16, there's a 40, which is 5/8. Yeah, in case math isn't your thing. Math is probably your thing, honestly. Or at least that kind of math. Here's a reconstruction of those two. So here's the little prong at the top. Here are the spiky little edges. That's the one we were just looking at. And here's that long, skinny one. All have roughly 8-fold symmetry, roughly, but it's probably bilateral symmetry. And at least in the early ones, they may have comb rows, and these are thought to be the comb rows down here, but they definitely have a skeleton. Yeah. And that is something that no modern ctenophore has, like this guy up here. So our little inspiration animal for today. So, yeah. Something new. Because remember, in evolution, you start out with a group that has basically a lot of possibilities, and you tend to get a lot of flexibility, and we call these crown groups. And these are just like ancestral groups from which multiple other branches will spring. Those branches usually occur by, essentially, it's like selecting some of the traits of the ancestor. It's not like they're really adding much of anything. They'll duplicate some genes and have a double version of a biosynthetic pathway, and maybe it'll go like one step further, like it'll add an extra carbon to your cholesterol molecule, something like that. But not a big change. They call these guys sclero, which means hard part, Ctenophora. So these are the armored ctenophores, this is how they think they are related to modern ctenophores. Then down over here, this would be all of the rest of animals would be branching off there. So these would be a sister group in the current reconstruction to literally everything. Okay. These are some other things that may be ctenophores and are a little harder to know for sure. So Shangwangia was originally proposed to be a cnidarian, and that's what I introduced it as in the Chengjiang lecture. That's not going to be on the test. This is just an area where reinterpretations, like moving it from one phylum to another, are actually happening. And the thing with crown groups is if they are the root of multiple phyla, then you can't really put them in any one phylum because they don't really go. Or if they're a side branch that is related to some phylum, you could put it in its own phylum, or you could just say, you know what, this is a crown group. Taxonomy kind of breaks down. Taxonomy relies on a lot of evolution having happened, and it's a way to separate things out that are at the end of the evolutionary path. The further you go back in the fossil record, the more that kind of falls apart. Yeah. So, yeah, here's a reinterpretation of this creature, which I think has 16-fold symmetry, as a ctenophore with a sort of cup down here at the bottom, a little bit of armoring on the sides, which is right here, and then these little fronds that stick out, and it's Cheng-Jiang, so you have excellent preservation of those soft tissue fronds with little pins coming out of them. That's what they call them. So. And here's another organism, Daihua Sangyong, and it looks like kind of the same thing as this, but instead of being, upright and together, it's got smashed like that. Or it was living and it was a little flatter, or it just, crushed like that. But you can see along there what appear to be something like a comb row. And it could be comb rows. These could be some sort of ciliated tentacle. They're probably for catching food. That would make the most sense. And if that's the case, then what you see here in modern ctenophores, these old, maybe food catchers have been repurposed as a movement device. These are the chief propulsion of modern ctenophores. And they eat usually by either catching prey on their tentacles or by actually grabbing and engulfing and digesting prey. The coolest videos about these on YouTube, which you can search for, are jellyfish, comb jellies, in fact, eating other comb jellies. Yes. Ctenophore on ctenophore violence. Yeah. Where'll it stop? Stop the madness. Here's another one. Yeah. It's got a big prong that's stuck in the ground. It's got a hard cup, sort of extensive, and there are the little tentacle things coming at the top. So the idea of, and it's also Cheng-Jiang, the idea of this paper was that, hey, all these things look like they may be part of the same group. And these are all the things that you see joined up right here in the Ctenophora. And here's another one from the Burgess Shale. And this one has what appears to be like a little holdfast, a little root. Although it's questionable whether that was actually serving that purpose or whether it was just that sort of shape. But if you reinterpret it in that way, you can say, wow, okay, well, those could be the little comb rows or, feeding apparatus. And it's got a hard case that goes around it. And there's really nothing like that today. It wouldn't be an echinoderm, because an echinoderm would have basically calcium carbonate. It's sort of a bubbly, airy, light calcium carbonate. Bones running down these tentacles and running down the center, and it's just hollow here. It is not that. And they would have calcium carbonate mineralization in little chunks. And what we're seeing are big, long sheets of what may be calcium phosphate, although with Cheng-Jiang and Burgess Shale, you never quite know, or it's hard to be sure. But yeah, here are the little combs. Look at that. Yeah. So maybe they are. Here's a Galliactina, which is probably in that same group as the armored ctenophore-like things. And so all of these, they think, were probably stuck into the ground with their hard part actually serving as an anchor point, and then they could close, perhaps, to avoid, dust and dirt being thrown around in the water when there's waves, and they could open and feed with their little tentacles. Yeah. So that's weird, but this is Cambrian explosion, and there's a lot of experiments that I don't know. Maybe they did go somewhere. It's just they didn't go somewhere that's easy to interpret. because there's a lot of filter feeders that work that way now. Things like brachiopods do, but everything is different other than that, and so you would be very brave to say, "Oh yeah, these are directly the ancestors of brachiopods." Yeah, because I don't know. I don't think the evidence is there. Here are some other things that appear to be ctenophores from the Burgess Shale. Some of these have far more comb rows than you would expect on a normal ctenophore. And usually it's a multiple of 8, if I remember correctly. But here are each of the little rows, and so they're showing little expansions of some of those. Here's one with a real hard cup at the bottom, so this may be one that lived buried infaunally, at least semi-infaunally. And here are some others, and look at all those. 1, 2, 3, 4, 5, 6, 7, 8. So that might be one of the ones that has 40 comb rows on it. There's a lot. You can count them on your own time. So there we go. This is the reconstructed life position of these creatures. And there's a really good fossil record of them at Chengjiang. But remember, Chengjiang is creatures that were buried in the ground, getting covered in a landslide, and they can't move because they don't move, and so they become wonderful fossils. Every time a bad thing happens, you get a wonderful little fossil bed there. And we think they were either pushed into the anoxic zone or may have lived in the sort of suboxic, like low oxygen zone, and we're just filter feeding away. And you do get animals, like proper metazoan animals, living in almost completely anoxic zones in the bottom of the ocean sometimes. It's rare, and when people find it, they still get kind of excited, but definitely a thing that can happen. Weird as that seems. So here's some other fossils. Some of these we've seen before, some of the others just to have a look at them. So these are ctenophores through time, and we have a lot of ctenophore-like things from the Cambrian. And then the fossil record kind of goes dark. You get that one in the Hunsrück Shale, which is Devonian. So think like the middle of the Paleozoic. And then they basically go dark again, and then you get the modern ones swimming around in the ocean. So they could be considered like living fossils in that we have fossils of things like them that go way back. And they never really left. They just stopped being fossilized. So there's something about the early conditions around the time of Chengjiang and Burgess Shale and Hunsrück that just happened to preserve soft-bodied organisms like these that usually were probably there, but did not leave a good fossil record. Here's a little ctenophore egg that they actually etched out of one of these little nodules. I think this one's preserved in phosphate, and if you crack it open inside, they actually sectioned it. There are little tiny comb rows in there, and there are 8 of them. And so, it actually looks a lot like a modern ctenophore embryo, and so they think that's what it is, although it may be an embryo of something like these guys, given the time. Because those, presumably, if they're related to ctenophores, which the other tree suggested, then those would probably have ctenophore-like embryos. Yeah, almost certainly. Here's some more beautiful ctenophores. This is that one with the long rows and the short rows, and here it is if you were looking down from the top of it, and a bunch of other organisms. These are some of the ones that probably lived halfway buried. These guys are probably pelagic. They're probably swimming. This one, fasciculus, I thought was epibenthic. It would just sort of sit on the bottom, but not be rooted down. And various others that have little comb rows. These, I think, are the same species as that. And there you go. Here's our little ctenophore embryo if you want to see it up closer and count. There you go, 1 through 8, the little comb rows. So, what else about ctenophores is neat? So these things have effectively a nerve net of things that work like nerves in that they carry impulses, electrical impulses down them, but they don't use any of the same neurotransmitters that we find in the rest of the tree of life. So you get as low as Cnidaria, jellyfish. They have nerve nets, but their nerves run on the same neurotransmitters, at least some of them, as us. And I don't remember if that's acetylcholine or GABA or what, but yeah, serotonin possibly. I know there are some very primitive serotonergic nerves. These guys will use it's glutamine and glutamate and aspartate amino acid as their neurotransmitters. So they kind of function similarly, but they're the only things that have this. So the idea is that either they are using a common shared set of equipment that has been lost from sponges and lost from placozoans, but retained in higher animals. And they just happen to evolve a different neurotransmitter, or they reflect and either they independently like invented nerves from components they had lying around, or they reflect an earlier version and a newer, better neurotransmitter, better in some intangible way, replace that in these lineages. I don't even know if it's better. It's just things get locked in, and you do the best with what you've got where you are. I think is what you can say about evolution. So there we go. We got to the end. It took a while, but it was worth it, I'm sure. Unusual aspects of Cambrian ctenophores: armor, spines, hard parts, way too many comb rows, statoliths, just like normal. Little comb rows, kind of like normal. Maybe they're being used for filter feeding, though, instead of for movement, so that's a little bit different. Yeah, and we have no idea whether the early ones would have also had glutamate transmitted nerves. And I don't believe they have. I don't believe anyone has published any papers examining the structure of the nerve net in these, because it's like a distributed brain that sort of runs around the body. It's a little bit concentrated at one end, which I think would be up here. I believe around the anus. Yeah. But yeah, it's serving the same function. It's just not all in one place, which, if you think about it, makes a lot of sense. I mean, we put all our brain meat up here, and then we go play football and drive motorcycles and stuff like that. And if bad things happen to this part, then yeah, you're kind of, it's kind of over. So I don't know. Yeah, not the worst idea. Nerve net instead of a big chunky meat brain all concentrated in a little bobble that sits kind of out from the body where all sorts of bad stuff can happen to it. But hey, yeah, I didn't invent this stuff. I'm just dealing with the consequences. What was their ecology? Epibenthic or low key in faunal, just like a little part stuck in the ground and then the rest filter feeding above. Let's see, how did that contribute to preservation? If you're on the ground or in the ground and the landslide comes, yeah, at least you're not going anywhere. And then it's a question of what minerals are present, what bacteria can help to precipitate them. Does something come in and eat you or not? Is it anoxic enough to sort of prevent that happening or select certain types of bacteria? And those are the questions you got to ask with any fossil. It's a miracle when anything gets preserved, and yeah, these that's what these guys are. How are they similar? Yeah, we talked about that. How are they related to other metazoa? We think they're a sister group to all the other metazoa, which is pretty crazy, considering that the bottom two branches of metazoa, the Porifera and Placozoa, essentially have no organization. Placozoa has these distributed, it's like a little flat blob, and it's got distributed digestive patches on it. And so it's thought to like sit on something and sort of absorb it. Yeah, it's just pretty crazy. But yeah, very little organization at the body level. Similar thing with sponges. You get a little bit of organization around the water flow, but it's almost as though the water flow is creating the organization. And no overall body plan on at least some groups of them, although hexactinellid sponges have really nice symmetry sometimes. Yeah, things like Hox genes, I believe, are not found here, although different homeobox type genes are found. So there you go. All right. And why is there uncertainty about the biominerals used by the Cambrian ctenophores? We totally covered that. All right, I think you got this. Thanks.